Good evening, folks. I have discussion E for you. It's an extra tonight. Okay, we're going to discuss. Here is the title: The Quiet Turning. How the Aging Brain's Immune System Rewrites Itself in Midlife. Okay, so here we go with tonight's discussion E for you. Give me 1 second, please. Okay, for decades, the brain's immune system was thought to be a model of consistency. Microglia, the resident immune cells seeded before birth, were believed to persist throughout life, quietly tending the neural landscape. They pruned synapses, cleared debris, and maintained the delicate equilibrium that makes memory possible. Their presence was taken as a given, a biological promise that the brain's internal guardians would remain steadfast. This belief shaped the narrative of aging. If microglia were lifelong companions, then cognitive decline must be slow. Linear, inevitable, a gentle erosion, not a sudden break, but new single cell research from human hippocampal tissue reveals a different story, one with thresholds, ruptures, and midlife transformations. It suggests that the brain's immune system does not simply age, it reorganizes. And in that reorganization lies the earliest seeds of neurodegenerative disease. The midlife pivot, somewhere between 50 and 75, the brain crosses a biological threshold. It does not announce itself. There is no tremor, no confusion, no forgetting of a name, but deep in the hippocampus, the microglia that have been present since embryogenesis begin to disappear. Their gene expression signatures fade. Their hormostatic programs weaken. their identity dissolves, and then quietly they are replaced. The newcomers carry transcriptional fingerprints more typical of peripheral immune cells. They are more inflammatory, less specialized, less attuned to the subtleties of synaptic life. They arrive with different loyalties shaped by the body's immune history rather than the brain's developmental choreography. It is as though the brain, once a sealed sanctuary, has opened a side door. This is not aging as a drift. This is aging as recognition. The collapse within. At the same time, something more profound unfolds inside the nucleus of aging brain cells. The 3D architecture of the genome, the loops, folds, and boundaries that determine which genes can speak and which must remain silent begins to erode. The chromation loops dissolve, regulatory neighborhoods blur, identity becomes unstable. This collapse is not metaphorical, it is structural. The scaffolding that keeps a cell's purpose intact begins to buckle, making inflammatory genes easier to activate and protective programs harder to maintain. The genome loses its choreography. The cell loses its compass. This erosion of genome architecture may be one of the fundamental features of human brain aging. An internal unraveling that precedes symptoms by decades. You can explore this further through genome architecture changes. The broader weakness, the blood-brain barrier, long considered one of the body's most vigilant borders, also shows signs of strain. The cells responsible for maintaining its integrity decline. Tight junctions loosen. The boundary becomes permeable. Signals from the body, stress hormones, inflammatory cytokines, metabolic bioproducts gain access to the brain's interior. The newcomers respond with alarm, amplifying inflammation. The feedback loop accelerates. The hippocampus, ground zero for memory, becomes the most vulnerable site of this transformation. It is no coincidence that Alzheimer's pathology begins here. Aging, it turns out, is not a gentle unraveling. It's a series of thresholds, and the brain crosses one of its most important thresholds in midlife. The human consequence for clinicians, caregivers, and families. This science is not abstract. It explains why a person can seem perfectly well at 60 and profoundly changed at 70. It explains why memory loss feels sudden, even when its roots are decades deep. It explains why the work of palliative care often begins long before the world dementia is spoken aloud. It reframes the lived experience of aging. The sudden irritability that emerges in late life, the subtle shifts in personality, the quiet withdrawal from social life, the early cracks in executive function. These are not simply psychological changes, they are biological consequences of a brain whose immune identity has been rewritten. The gendered story. Emerging research suggests that women may experience more pronounced midlife neuroinflammatory shifts, potentially linked to hormonal transitions. Mitochondrial vulnerability and immune system remodeling. The new study does not resolve these questions, but adds weight to the idea that gendered risk is not a mystery. It is a biological trajectory. For health system leaders, this matters. For public health, it matters even more. If midlife is the pivot point, then prevention must begin earlier, perhaps even before age 50. The new narrative we must tell. The old narrative, microglia as lifelong guardians, the brain as sealed fortress, aging as slow decline, no longer holds. The new story is more dynamic, more fragile, more urgent. The brain's immune system does not simply age, it reorganizes. Its genome does not merely mutate, it loses its scaffolding. It 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 borders its borders do not quietly weaken, they open. It's a story with profound implications for neuroscience, geoscience, and public health. But it is also a story about people, about the patients you care for, the families you counsel, the systems you lead. Midlife is not too early not to intervene. It is the moment when the future is still malleable. The invitation. This new science invites us to rethink aging not as a slow fade, but a series of biological inflection points. It invites us to imagine interventions that restore microglia homeostasis, strengthen the blood-brain barrier, and stabilize genome architecture. It invites us to consider aging not as a destiny, but as a modifiable process. Most of all, it invites us to tell a new story, one that honors the complexity of the aging brain and the humanity of those who live within its changing landscape. The path forward, a full reframing of brain aging requires a new biomarkers that detect microglia turnover in midlife, longitudinal studies that track immune cells identity across decades, preventable or preventative interventions that began before symptoms, health system strategies that integrate brain immune aging into chronic care planning, public health messaging that shifts dementia from an old age inevitability to a midlife process. This is not merely scientific process. It is a cultural process. It changes how we talk about aging, how we prepare for it, and how we care for those who live through its transformations. Aging, the aging brain is not a statistic or organ. It is a landscape in motion, cells departing, newcomers arriving, borders shifting, identities dissolving, and reforming. Midlife is not a quiet plateau, but a hinge, a turning point, a biological pilot twist. To understand aging is to understand this turning. To care for aging people is to honor its complexity. To lead health systems is to anticipate its consequences. And to write about aging, as you do, Felicia, is to give language to the invisible transformations that shape the final decades of a human life. Okay, guys, that is it for tonight. I will see everybody tomorrow for shows. Until then, Gator Wellness, have a good evening, folks.